Good afternoon, everyone. Welcome to the December 12th webinar series of the IMAG-MSM Working Group on Multiscale Modeling and Viral Pandemics and the Global Alliance for Immune Prediction and Intervention, or GLIMPRINT. This meeting is organized by myself and James Glazier, and with a lot of help by Jim Sluka, as well as Lorenzo. If anybody has ever any questions, suggestions for improvement, feel free to contact us directly. We also appreciate if you help us spread the word about individual webinars, but also the working group and GLIMPRINT in general across our different social media channels, ranging from Slack to X, LinkedIn, and also YouTube. All of these webinars are being recorded, live streamlined to YouTube, and also archived there as well. If you have social media, social media posts, please help us again retweeting them or re-Xing them on X, and help us grow the community and awareness of about multiscale modeling. If you're working in the space and have any recent publications, please feel free to email Jim Sluka at jsluka@iu.edu. We'd be happy to promote those publications on our website. We have tens of different publications that were co-written by many members of this community. We can also post your job listings. So if you have any for your institution or any relevant job postings that you feel need attention, please feel free to, again, email either Jim, or feel free to go to glimprint.org to submit them there. Currently available positions in the field of modeling, multiscale modeling at the University of Florida. There are several faculty positions in the Laboratory for Systems Medicine, headed by Reinhard Labenbacher. Feel free to either check out the website or email Reinhard with additional questions. At the University of Nebraska-Lincoln and our Digital Twin Innovation Hub, my team is currently expanding, and we're looking for several staff positions, modelers with experience in multiscale modeling, agent-based modeling, and we're also looking for software engineers. Jason Shoemaker at Pittsburgh University is also looking for PhD students and postdocs to work on modeling of influenza infection. So again, feel free to contact him directly. His email is jason.shoemaker@pitt.edu. A couple of announcements of grant opportunities in the space. ARPA-H has a Catalyst RFA that's available at the moment for toxicology and physiology modeling. NSF, NIH, and FDA all have been very recently developing and making available new funding opportunities in the field of digital twin technologies. NSF, they all have invested over $6 million across different projects already, and there are many RFAs available, active RFAs available right now, including this NSF one that's more software oriented, developing open source software tools. NIH, NIAID in particular, has a notice of special interest for systems modeling of infection and immunity across biological scales. This NOSI applies to can be applied to standard R01 type applications. And under this NOSI, the investigators would work with the U54 Center of Excellence for Systems Modeling of Infection and Immunity across biological scales. So there is expected partnership. I am not sure if the winner of this RFA of the center has been announced yet, but nonetheless, there will be an expectation of collaboration. Workshop announcements. There will be a seventh workshop on virus dynamics in France next October 14th through the 16th. We will have a steering committee tomorrow at 11 o'clock Eastern time. Upcoming presentations. Martina Iullini from University of Milan will be speaking on January 9th. And then we have several open slots still in January and February. So if you would like to nominate yourself or anybody in your circles, please do so. And you can also suggest speakers at this URL, and then Lorenzo will work with you to schedule folks onto the calendar. It's my great pleasure for today's webinar to introduce Valeriu Damian, who is a Senior Director of Systems Modeling and Translational Biology at GSK. And thank you so much for being here, and we look forward to your presentation, Damian.
So first of all, thanks for the invitation to present on digital twin approaches in quantitative systems pharmacology. So for my talk today, I will talk first about QSP a little bit, just in brief around the impact about model building, modeler approach for building a QSP model, and then discuss a little bit about QSP workflows. And the reason for doing this is because I wanted to talk about virtual population and then the digital twin concepts in the context of virtual populations. And then I'm going to be using some examples of using digital twins for both population inference and for patient benefit with an example in bone marrow transplant. So to get started, this is a slide from a paper from Piet van der Graaf in 2013, basically looking at in order to get confidence in proof of concept in a clinical trial, you need to have confidence in compound that requires, the good three pillars of target exposure, engagement, and target modulation. But the part that we are mostly interested in is on the confidence in target that requires essentially understanding the systems pharmacology, and that's effectively what quantitative systems pharmacology is all about. And for the purpose, at least of this presentation, I've been defining the quantitative systems pharmacology as a mechanistic mathematical modeling approach to predict the effect of target modulation on clinical outcomes. Now, QSP has been around for a while, but in the last, several years now, it has gotten bigger and bigger and having more impact. This is showing a recent update from a landscape from the US FDA that shows the number of submissions that include QSP modeling. So you can see that it has grown over the years quite a bit. And if you're looking at the number of these submissions that are separated in various disease areas, quite a few of them are actually in oncology. And obviously, you can anything that has connections with the immune system will probably apply quite a lot in immuno-oncology in particular. So what I wanted to start here is show an example of a platform model for immuno-oncology that I built while I was at GSK, together with my colleagues Lavlina, Alap, and Roy. And the idea here was to actually try to put together a model that is comprehensive as a platform model for immuno-oncology. In order to build this, we didn't want to build a model. We wanted to build a framework in which the models can be easily developed and extended. So we started with cell modules. The cell module basically tells what a particular cell type does, in what tissue, in what tissues does it exist, in what states does it exist, and for any kind of cell transition, what are the effectors that facilitate the transition of a cell from one state to another. We added effector modules. So this is looking at the various effectors and having an understanding of which cell types are producing that particular effectors. Then we added, obviously, compartments, and the natural ones would be the blood, the lymph, and the tissue. And then we started expanding the tissue compartment. For instance, for tumor purposes, we wanted to split that compartment into 3 regions at the very least. What we did here was, having in a tumor microenvironment, a normoxic, hypoxic, and a necrotic region. We have all the different cells in the various compartments moving about from between these, and basically keeping track of all the cell types that have been included in the cell modules. Putting all of this together produces a core model, but that's not sufficient, because when you start looking at the cell-cell interaction, you want to include a little bit more detail depending on the drugs of interest. For that purpose, we basically defined templates, cell-cell interaction templates, modules. For instance, here I was looking at how an APC cell interacts with a T cell and activates the T cell. And that basically happens by forming this immunological synapse, and then sort of triggering regulation signals inside the cell to activate the T cell. And some of this interactions between the APC and the T cell happens through receptor ligand interaction. So if that's something that was of interest, that was expanded with a particular template. So the way that this model was built was that anytime in a reaction that was a cell-cell interaction, or it was mitigated by a cell-cell interaction, you put a tag on that particular reaction. And then that tag allows you to basically go into the appropriate template and expand the model as appropriate. So this allows you to actually build the model sort of dynamically as you need it, and include as much detail as needed. So this model, when you put it all together, we spent time for one purpose to actually put this diagram together. You see all the explicit cell-cell interactions, cytokines, chemokines were excluded from the diagram. That's how it looks like. And this model was actually put together around 2018, 2019. You can see the timing for developing these kind of modules, putting the framework together in 2019. We started calibrating the model initially for the healthy state, calibration for cancer patients, and then from 2020 onwards to now, on one hand, we expanded the model to include more receptors. You see receptors A, B, C, and so forth. We expanded the model to incorporate validation of different cancer types. And then we used it for evaluating combination therapy, to evaluate novel targets. We used it to terminate programs where it was appropriate without actually having to go into a clinical trial. And we had submissions to the FDA. The part that I wanted to spend a little bit more time is this issue about a virtual population and virtual population generation. And for that, let me talk about QSP workflows. And in order to build a QSP model, we've seen already, you start with defining the intervention, the outputs, gather the data that's required, separate calibration and validation data, define the model, and this is the part that I spend a lot of time discussing so far, trying to build it from different pieces, and then calibrate the model based on usually in vitro data. But that's not sufficient. This is basically just calibrates the basic structure of the model. You still need to calibrate it for individual patients and for populations of in a particular trial. For that, so let's look at what it means to evaluate new treatments using virtual population approaches for a QSP model. So I represented here first the outcome space. So these are outcome space for an existing trial. These are real patients, and this is where their outcomes sit in the space. And this is the population level outcomes for this trial. Next, we have a QSP model, and therefore we can define the parameter space in this QSP model. These are virtual patients in this space, and these virtual patients we can use the QSP model to simulate. Now, in some cases, the simulation is going to get us into the feasible outcome space. In some cases, like here on the top, maybe not. So this blob here defines the plausible virtual patients. As we have it initially, if I flip between these two, you'll see that you can generate these virtual patients, and the distribution of the outcomes may not necessarily match the distribution that you've seen in the clinic. So for that purpose, to match these two, you keep sampling the plausible virtual patients until you get a match between the distribution of clinical outcomes and the distribution resulting from taking virtual patients and simulating them through the QSP. Once you have something that matches perfectly the existing trials, you have essentially a calibrated, fully calibrated model, and you can start making prediction moving forward. For that, you look at the outcome space for a new trial. You are using the QSP model to predict in this space the outcomes for this new treatment, for instance. And then you basically look at the population level outcomes for this new trial. All right, so we have now the basic virtual population approaches. Looking at the different elements that need to be accomplished to actually build something like this, you have the first step of creating virtual parameters, selecting the parameters that define a virtual patient, generating the virtual patient, selecting the plausible virtual patients, calculating a population, validating it before you can go in and apply it, and perhaps reapplying it. So let's look first at selecting the parameters that define a virtual a patient. You can do this either by expert opinion, local sensitivity, global sensitivity approaches. There are many algorithms, but there are also quite a number of challenges. The dimensionality of these is quite high, and even though most parameters that you have some good ranges from in vitro data, there is still some uncertainty in those that differentiates one patient from another. And there is correlation, unknown correlation issues with these parameters. You can't really identify them. The plausible parameter ranges, they're not always clear. But on the plus side, it provides a way to correlate these parameters with genotype. Generating the virtual patient and selecting the plausible virtual patients, it's a little bit tricky, because first of all, you need to do sampling of the parameter space. Again, many algorithms for doing that. But once you select a sampling from the parameter space, you need to calculate biomarkers. You need to look at the different time points. You need to look at different trials, different doses perhaps, or completely different trials, different treatments for the same patient. And then once you do this, you need to filter the plausible patients. And in some cases, in our experience, it's only less than 1% of the samples that you try lead to actually viable virtual patients. And basically, it's a computational issue. And then, you want to improve it, perhaps using machine learning, using surrogates. I've discovered to my, in some of our trials that the parameter space is definitely not convex. If it was, you could just take the middle of two randomly selected virtual patients and generate a viable new virtual patient. That doesn't work all the time. Generate distinct phenotypes. There's no point in generating a huge number of virtual patients that are all very, very similar. And then finally, digital twin approaches that could help in this space. More about that later. So in order to look at the calculating a virtual population to match the calibration data, there are many approaches, from prevalence weighting to Monte Carlo type approaches. But sometimes when you do you generate the virtual patients, you may not cover fully the distribution of the observed outcome. So you need to try to generate virtual patients that sort of fill that space properly. And one of the challenges here is that the process of generating virtual population is actually quite difficult, quite computationally expensive. And if you want to maintain the model, if you make changes to the model, then all of that calculation needs to start all over again. I haven't yet identified algorithms that would allow me to merge, to transform a virtual patient that was defined in one model in a model that has gotten through some updates. All right, so I'm moving now to digital twins. So we looked up to this point about how to define a QSP model, how to define a workflow for QSP models. Talking about digital twins, if I look at the definition, a digital twin is a digital model of a product, system, or process that serve as effectively indistinguishable digital counterpart for practical purposes. So paraphrasing this, in order to have a digital twin, one needs to specify the system or the process and the purpose. So let's look at some examples. If I want to build a digital twin for a disease, for the purpose of predicting the disease X population response to therapies, that is effectively what we've been developing as a QSP model for disease X. So a digital twin for disease X for this purpose, it's effectively something we've all done before. A system or a process, a digital twin for a drug Y, for the purpose of predicting population exposure for drug I, that's effectively a PBPK model for drug Y. A digital twin for a patient Z and drug Y, for the purpose of predicting individual exposure for that patient and that drug, it's a PBPK parameterized for the patient, for the drug Y parameterized for the patient. This is actually something that's used for therapeutic drug monitoring. If again, I'm looking at a digital twin for a patient Z and a disease X for the purpose of predicting the individual response to therapy for that patient, that's the QSP for a disease X parameterized to match patient Z response. And this is basically the one that I'm going to focus mostly about. The other ones are just as examples. But looking at this, you can see very clearly the top two look at population benefit. So it benefits the population. You are using it to make population inference. And the last two are looking at examples that are meant to benefit patients. So this allows me to classify the digital twin application in QSP based on who benefits the most out of the approach. At the patient level, so for patient level benefit, you have personalized treatment, picking the right drug target, dosing schedule. You can use it for bedside application, for rare diseases, for personalized risk-benefit assessment. When you are looking at the population level, you have better approximation of population variability, better coverage of phenotypes, increase the trust in population level predictions, characterize perhaps sparse populations. So let me start talking about the population first, this application. And the reason for it is, there's been discussions about should we be using a digital twin approach or a virtual patient type approach. So I've shown this that in a previous slide, looking at the outcome space and trying to build virtual patients and digital twins in that context. But my claim is that all digital twins are virtual patients, but only those virtual patients matched to a real patient are actually digital twins. So if I look at it in the outcome space, we have a real patient represented there. In the QSP model, we have a QSP parameter space. We have virtual patients in the QSP parameter space. We can model them and project them into the outcome space. Some of them are viable, some of them are not. The plausible ones are in the space that I've identified. There usually is a reference virtual patient that actually corresponds to the center, the median of the outcome. And then for digital twins, digital twins are essentially plausible virtual patients that, given the same baseline and treatment as a particular patient X, they predict the same outcome as observed by patient X. So with this in mind, let's look at virtual cohorts. So I'm talking now about the cohort of virtual patients. So I selected a number of virtual patients in here. I project them in the outcome space. But when I look at the distribution, I see that the distribution of these patients are very different from what perhaps I observed in the clinic. I may be missing phenotypes. I'm just not matched the distribution, or I may have too few virtual patients. Put more virtual patients as a solution, and then do all of the MCMC or prevalence weighting approaches that I've discussed earlier. That would get me now to match this distribution. Now, there is a second solution here. I can start with real patients that were initially used to generate the distribution. I can build for each of these real patients their corresponding digital twins, and I can use the digital twins for any further inferences. This all works good, but the question is now, I have two alternatives, the virtual patient approach, the digital twin approach. How are they different? And you can see in here, the actual virtual patient digital twins are different. In the parameter space, they produce different outcomes, but they match in terms of the distribution with what has been observed clinically. So as a comparison, if I have, when do I use a virtual patient approach? When do I use a digital twin approach? If I have only population statistics, which would be the case if I take my information about what happened in a particular clinical trial from literature, I usually don't have patient level data. So if I have only population level statistics, I'm kind of stuck with using virtual patients. The advantage here with a virtual patient is I can incorporate data from many trials. I can generate as many VP as needed to describe properly the distribution. Digital twins approach, require patient level data and enough patients to describe the distribution. However, the inference based on digital twins it uses real patient data, so it has a little bit more trustiness to it. Obviously, the outcome distribution is easily matched. I don't need to do all of that prevalence weighting because it's kind of given to me already by using a real population. And we can link the outcomes in the QSP model with the genotype, which again is quite interesting. We can also merge the two approaches by seeding the virtual population with digital twins and expanding with virtual patients. An example of using this, it's a paper from Genentech on a phase 1 study. This was a paper that was awarded at ACOP an award, systems-based digital twins to characterize dose response in a bispecific antibody. Looking at this, I'm actually describing what they have done in a diagram similar to what I've presented so far. So essentially, they have in the outcome space the phase 1 patients, not that many, and each patient there, or several, is a relatively small number of patients that received different doses. For each of those patients, there is a digital twin that's been generated, but they haven't generated a single digital twin. They generated a few, they call it individualized virtual patients, or I would say approximate digital twins of this one patient here. And they've done this for all the different patients in the trial. So essentially, if I look at patient one, you have 25 digital twins for it. You calibrate based on treatment one, but then you can use and predict how this particular patient is going to behave under several other treatments. Patient 2 received perhaps a different treatment, but you can predict how it's going to behave all across, which means that at the end of the day, you not only have calibrated these virtual patients, but also have a population which is quite significant to actually make prediction across all of the different treatments. So that basically allows the in this paper to increase the confidence in their prediction. Moving forward, I wanted to discuss the digital twin application that are benefiting at the patient level. So let me describe the bone marrow transplant, and looking at the definition above there, for digital twin, it's the base. A better digital twin is one where that updates based on real-time data. And this is exactly the point of a digital twin that I wanted to focus on in this example. So in bone marrow transplant, this is days. This is the transplant day. Prior to the transplant, there is potential days for chemotherapy. After that, the neutrophil count actually achieves a minimum, and then after that, the neutrophil count recovers. And obviously, what you want is to actually not have too long of a period when the neutrophil count is low. But on the other hand, you need to be low enough to allow the cells that you are injecting, transplanting, to actually take on. So the question that I'm asking here is basically, how much data do we need about a particular patient to build a digital twin? So again, if I want to build a digital twin for the bone marrow transplant to predict population level neutropenic state, that's the QSP model for bone marrow transplant. But if I look at predicting the best chemotherapy that for that patient, that's therapeutic dose monitoring. And further, if I want to predict the response for patient Z to administer optimal rescue treatment that happens here after the transplant, then this is something that that's something that I'm going to be focusing on. What kind of treatment would be best to rescue this particular patient? So these are two different concepts here: usefulness of a digital twin versus accuracy. So let's look at it in the context of a bone marrow transplant. This is start of treatment, end of treatment, and this is prediction accuracy. As I get more data for a particular patient, I would expect the accuracy of the models to increase. At the same time, if I start, for instance, even before the treatment start, the best I can have is a generic digital twin. That it's not specific for a particular patient. It's leveraging population level knowledge that existed for bone marrow transplant. If I do it at the start of treatment for a particular patient, it means that I'm leveraging data from the patient's medical history to calibrate a digital twin. At the other end of the spectrum, I have what I would call a true digital twin, which is accurate. But at this point, the usefulness of it for the patient starts going down, because in the very beginning, it would be great if I could make a prediction. But on the other hand, if I finish my treatment, it may be too late for the patient to make any kind of changes. So there is a sweet spot in the middle, what I call the useful digital twin, which balances the accuracy of the model, the accuracy of the predictions, and the amount of data that I require to build such a model. So this is the model that I put together, the QSP model for bone marrow transplant, and it's based on a couple of literature models that were already available. This one looks at the host cells, and these are proliferating cells for the host, and then a couple of transit cells ending in the blood. This is the absolute neutrophil count, and then the neutrophil counts contribute to GCSF production. You add the donor cells. The donor cells basically undergo a very similar process. The chemotherapy has an effect in killing the cells and in preventing them going through this maturation cycle. The GCSF here, the GCSF effect basically promotes the maturation of the neutrophils. The host and the donor immune cells, basically looking at the set of immune cells of host type or donor type, would basically try to, the host immune cells would basically try to kill the donor cells. That's why you kill them first, essentially. And then you are looking at the chemotherapy, and in particular, busulfan. So busulfan is one of the chemotherapy agents that's used to deplete the neutrophil counts. And for that, the PK, it's actually done as in a therapeutic dose monitoring approach as a function of patient body weight, age, and so forth. And it has two effects, a direct effect and a delayed effect. The direct effect contributes to the proliferation and maturation of the neutrophils. The delayed effect contributes to the killing, essentially, of neutrophils. And then the other chemotherapy that each patient has received is done in a very simplistic way. The data set that I have to work with was 146 patients that were generated data was generated at UCSF over a number of years. Ages of patients from 0.2 years to 28 years, significant variation in the body weight, many reasons for the transplant indication, different types, different pretreatments. We used therapeutic dose monitoring for busulfan, GCSF as a rescue after transplant, and we measured daily neutrophil counts. The published neutropenia model and this data was used to build a QSP model. You've seen what the QSP model looks like, with all of the different elements. And then looking at the digital twin workflow, and I'll spend a bit of time describing this. So essentially what we've done here is for each patient, I use the patient-specific demographics, treatments, and dosing. And then for each day after transplant, I generated a digital twin population that is compatible with the patient data up to that specific day, and then before transplant. And in doing so, I've used a Markov chain Monte Carlo type approach. So essentially what I have here in terms of digital twin population is quite a number of patients in this population. And I have 146 patients, 30 days after transplant. This is how many, 4,000 digital twin populations. And then I use this to predict the effects for the rest of the patient data. And then you go ahead and calibrate this and analyze the predictive power of the digital twins. Actually, this is a conceptual workflow. In reality, I've applied a little bit of a trickery to actually get things to run a little bit quicker. This is just for computational sake. We start with a QSP model parameterized based on published model. And I call this a digital twin for the disease. This is the knowledge that I have about the disease without any kind of knowledge of this particular trial or data set. I generate random samples from the parameter space, 100,000. For each sample, for each patient, using the patient-specific demographics, I simulate the neutrophil and store the results. This ends up with still a large number of simulations, but a little bit more manageable. I select the parameter set that minimizes the error across all patients. So this is basically a set of parameters that works for everyone. So it's not a digital twin for an individual. It's a digital twin for the disease in this particular population. Then for each patient, I select the parameter set that minimizes the error for that one patient. So this behaves like a digital twin for the disease in this particular patient. Up to here, I essentially have a digital twin for one patient. There is no uncertainty whatsoever. Then I basically start using this digital twin population approach, where now I go for each patient and each day after transplant, I generate a digital twin population by sampling from the pre-calculated profiles using the same MCNC approach. So this is basically by doing the pre-calculations, and this I basically save a few simulations, and then analyze the digital twin population approach. So let's look at how this shows in practice. So first of all, let's look at the raw data that we have, and you can see that among four different types of patients, there's huge variability. As you expect, the neutrophil counts is relatively, normal range, I would say, give or take. Probably these values here are not normal. The neutrophil goes down after the chemotherapy due to the effect of the chemotherapy treatment, and here it comes back up, hopefully with the donor cells. So the shape, it's clearly understood here, not a problem, but the variability is huge. Now, what I'm showing here is the results of the first three approaches without any kind of variability. The error bars that you see in these plots are coming by looking at this across all the different patients. So I'm just focusing on one of them, autologous, inherently immunocompromised. So this is the error, the prediction error that I get for a digital twin of the disease. So this is basically the error in predicting the behavior if I have essentially a single profile predicted for all patients. I improve on that if I create a digital twin that's now calibrated for this particular population. So this is not my initial guess by, if you want, a fitted and optimized single parameterization that works for all patients. This one at the bottom is basically a digital twin for the patient. So now I've basically picked up for every patient a different parameterization that's best for that patient. And you see the trend of improving this across all types. But the most interesting part comes when you talk about the digital twin population. So let me explain this first of all. So at the top here, we have the treatments. So this is basically in red is the time when the chemotherapy was given. This is a different type of chemotherapy. This is the transplant at time 0. And then in green, this is the time when the patient received GCSF, so the rescue medication. And this is basically the true digital twin population. So what you see in here, obviously the dots are the experimental data. The green region represents the quantiles 5 to 95 and then 25 to 75, and the median in the middle of the prediction from this population of digital twins. And just for comparison, I have in here the digital twin for the three things that I had before: the digital twin for the disease, the digital twin for the population and for the patient. So you can see here that with a true digital twin, I predict the profile reasonably well for this particular patient. This is patient ID number 7. Now let's look at the same patient, but I'm using just data up to day 7. Here I've used all the data. If I use data up to day 7, as you could expect, I predict okay up to day 7, but after that, the variability, the uncertainty in my prediction goes up. And that's what you'd expect to see if you haven't calibrated, if you haven't seen enough data for the particular patient. And you see this trend continuing for, a couple of other patients. This particular one, the true digital twin and the data shows that this patient essentially does not recover. So the neutrophil count stays at 0, whereas the digital twins that I had, for the disease, they predict that it will recover. Even if I go up to day 7 and use just partial amount of data, it tells me that the most likely scenario is that it's not going to recover, but there are still a relatively small probability in the predictions that I'm making at that point that the patient will recover. This patient does pretty well. Another example here where it does pretty well, and the same trend holds. So how can I now analyze this this idea that the more data I have, the better prediction error I'm gonna have, the more accuracy I'm gonna have. So this is what we've done with this. So we actually looked at the prediction error, and this is across days of the trial. This one is the pre-treatment digital twin. So this is just leveraging the patient medical data history, essentially. And you can see that I'm doing okay-ish, perhaps, in here. Now, why is that? Because this is the time when the neutrophil count is going to be 0 by design. So the fact that I'm predicting it to be 0, well, it's not a great achievement. The place where I'm not doing well, it's around the recovery part. Now, if I go with, on the other end of the spectrum, at a true digital twin, using all the data, this is the best I can do. And you can see that even there is a little bit of uncertainty, and there is a little bit of, it's not sort of balanced very well. But there are regions where I predict very well what's happening and regions where I don't. And moreover, you see that between day 21 and day 28, not much of a difference. So if I have data up to day 21, I can predict everything pretty accurately, or as accurately as I could. On the other hand, if I probably look at something where I have a little bit more usefulness to the approach, the useful digital twin, predict using data up to day 17. I still have a chance to make some useful predictions moving forward because I still have some days left, but the accuracy, at day 17 has improved tremendously. So the question is which data from the patient is the most important one? So what I've done here is I looked at, again, prediction error, and I looked at using data up until day 1, 2, all the way to 27 for calibration. So what you see here, and just focus on the mean for this. So the error is relatively, high, I would say, all the way up to day 13, 14. And so essentially that means that almost anything that I learn from the data during this period is not going to help me make better predictions. But moving from day 13 all the way to day 19, this is the time when I'm learning a lot from whether this patient is going to have a lot about the patient and his response to to the cell transplant. And after that, again, I'm doing a pretty good job. There's nothing much else I can do. Another way of looking at this is looking at digital twins, if you want for a bedside type application. So can I use this model to predict the ANC levels and days in advance? So this is looking across all the different patients, asking the question, if I know the data up to some day, can I predict next day? Can I predict next 2 days, next 3 days in advance? So as you would expect, the more you need to make a prediction in the future, the less accurate you are going to be. And if you want to predict the next day, that's going to be pretty accurate. The usefulness of this is that the model at this point can predict, what's going to happen if you give the rescue medication, you don't give the rescue medication, you give it at a different dose. So you would have confidence that it will predict correctly the next day, maybe the next 2 days, probably not as well moving forward to predicting 10 days. So hopefully I showed here some applications of digital twins in the context of QSP. I've looked at it in the context of usefulness for patients or usefulness for population. These are a couple of opportunities. By the patient level, you can leverage genetic information. You can leverage wearable technologies to incorporate in this. Incorporate uncertainty and usefulness and apply this digital twin population as opposed to just building a single digital twin. But there are challenges here. How comprehensive does a model need to be useful? How fast? Because if I need to run this on a supercomputer to predict what's going to happen tomorrow, that's kind of useless as well. And how accurate, again, this personalized needs to be useful for a doctor. The doctor already has the ability to figure out what needs to be given to this patient. When do I pass that threshold of actually helping the doctor as opposed to perhaps not providing any additional useful information? At the population level is basically dealing with heterogeneous populations, and it allows you to do with such populations. Again, genetic information, companion diagnostics would be something you can use here. And I think that it's useful to not oversell the digital twin approach. And understand the benefits, but also understand limitations and sort of apply it and use it appropriately. So with this, I'm going to stop and acknowledge this is work that I've done mostly while I was still at JSK. This is the folks that were in my team when I've done this development. These are in JSK in Viv, Joe, and Andrew. There is nothing would have happened on the neutrophil discussion without all the hard work and input from folks at UCSF, Beth, Janelle, and Jordan, and then folks in JSK IO team. This is the biologists that I work with there. And with this, I thank you very much, and I'm looking forward to questions.
Thank you so much for a great presentation, Valeriu. Any questions from the community? So maybe I'll kick off the questions. So you mentioned FDA approvals. Has your particular model been submitted to the FDA, or?
So first of all, there were submissions, not necessarily approvals. I'm not sure how many of them ended up in approvals. I mean, the FDA hasn't shared that information. And from the submissions that I have done with the model, there were a couple of them, and they were accepted. But again, for a QSP model, it's basically a combination between what you are asking. Is not that they approve the model, is what you're asking from the model. And is that sort of the only supporting evidence, or is it additional evidence in there? And in some cases, I would say depending on how ambitious you are with what you're asking. If you ask the model, for instance, to allow approval of a drug for a new indication without a clinical trial, that's a tall order. But on the other hand, if you just need to demonstrate that you can adjust the dose in a particular way based on the model, or justify why a particular dose would be better than other, then that might actually be done based on the QSP.
Okay. And what kind of data or information related to the model does the FDA expect, in terms of validations, parameters, and annotations or evidence that the model is, based on real data or convincing data? To me, it's been a black box, but it's something that I'm very interested in with what we're doing eventually.
So I have two answers to this. First of all, everything that you mentioned there needs to be disclosed to the FDA when you're submitting a model. All of the parameters, all of the equations, the model code, the justification for where every parameter has come from, as well as the data that you've calibrated with, and everything that you want the model to support, or the question that you want the model to be used for. There is a working group at this point that tries to put some guidance about how to provide a submission of a QSP model to the regulatories. That will provide perhaps more clarity. But at the end of the day, anything from validation to to model development and validation and everything, all of it needs to be included in there.
Yeah, I guess my where I'm uncertain is how much, because depending on how complex the model is, that's going to increase the scope of the validations, and is validating the model against 10 biological phenomena versus 100 versus 200. What is the sort of expectation, if there is one?
So there is one, and it's basically from a regulatory perspective, they basically look at, I need enough validation to be able to support the claim, whatever you want out of the model. The more ambitious you want with what you're asking them to do with the model, the more riskier that is, the more validation you need. And I can tell you that there are some efforts at this point of people that try to essentially keep maintaining a QSP model and updating it online with or online or in an automated way with new data that becomes available. So there are a couple of models out there that basically, when a new trial becomes available, they make sure that, first of all, you can simulate that trial, and if there are discrepancies, that you refine it. And this is one of the reasons why applying virtual population approaches is useful, because it's very rare that you see publication that gives you patient level data. Obviously, internally, you can have that for the trial that are sponsored by a particular company, and even then, it's actually quite challenging to get access to patient level data because of privacy concerns.
Hi, thanks for a nice talk. This is partially overlapping with Tom's question maybe, but I'm curious, rather than the FDA side, the clinical trial side, like, has there been a clinical trial that has tested whether model-based interventions are beneficial, like in this space?
Model-based interventions are beneficial. So this would be, let's say that you are using a QSP model to either identify a new target or to justify what a particular drug is going to be efficacious or not. And that's basically what some of those submissions are all about. And most of them, if not, well, a lot of them would lead to successful trials. I can give another example.
But has there been a trial already, or is that coming after FDA approval in some way?
I mean, the FDA would not approve anything without the trial. Right, so the FDA is giving you permission if you want to actually run the trial. And then once you run the trial, the approval for the drug comes only after you get the actual results. But the usefulness of the model is to justify why a particular dose might be useful and not, safe and efficacious for patients. I would give you a slightly different answer as well. And this is again from work that I've done in JSK. We had built a portfolio of models, QSP models in JSK. Give an example, we had about 20 QSP models developed and trying to maintain them as best as we could. And sometimes you see headline news that a particular drug has failed, company X invested billions and the drug failed. If we had a model at the time for the indication that was in that particular trial, we would try to simulate that that trial. And actually, in all these simulations that we've done, we were able to predict that that drug would have failed. Yeah, we've also predicted, positive results as well with our models. So the models are are quite predictive in that case. There is one example, actually it is from a few years back, where the FDA has approved a drug at a dose that hasn't been tested in human based on a QSP model that the sponsor company never submitted. It was something that was published. The FDA basically took the model, simulated it, figured out that's going to be safe, and then basically provided that approval. This is, if you want to look at it, this is for Naptara, a PTH receptor drug.
Thank you. Elena.
Hi, my name is Elena Zukova. I'm actually a research scientist at FDA, but I don't work on QSP. I work on medical imaging and AI in CDRH. I'm very interested in sort of whether any kind of AI can be used for some aspects of QSP or sort of making these digital twins, because my understanding is that in QSP you have equations that govern sort of everything, and sort of what do you think about it, and how would one, if one were to use AI, how would you validate that kind of model?
So probably I should have started with my first slide there. If you noticed, there was an image on the first slide with a image generated by ChatGPT. I thought that I have to give the title of the talk to ChatGPT, see what image it creates, and it was quite nice. But no, that's kind of a joke in there. There are a couple of places where AI and machine learning can actually help in this space, and I categorize them in two buckets. First one of them is basically I look at the workflows that I presented, and I want to improve the workflows here in different areas by using AI and machine learning. And one place is basically trying to derive some of the equations, trying to derive the structure of the model, trying to get let's say you actually work with a model as it is, trying to build a surrogate model that would allow you to identify plausible patients easier. And there is a lot more that can happen in that space with machine learning. So, but all of those are essentially. The other one that's quite important is, when you're building a QSP model, you're typically working with parameters, the space in the QSP model that are something that you typically measure, but not in the clinic. You're measuring in vitro settings. If you want to link with the genotype, that's a very difficult problem for a QSP model because you don't have the genes per se in your model. But having a digital twin type approach where you know the genotype of the patient, you know the outcome, you use the model to back calculate the parameters, then you can look at the parameters and the genotype and try to identify kind of correlations in there and see if they hold up moving forward. So that's again a place where machine learning can help. Now there is another angle to this second bucket. And this is actually a bucket that I've been trying to think about it. I basically challenge someone to say, if the problem is trying to identify, let's say, address a model-informed drug discovery development question, and you want to address that question, what dose to use, and instead of applying a mechanistic model, you say, forget about all that. I'm going to use it machine learning, only machine learning. Now, currently, you can't do that. There is not enough data to give you the granularity to give you the information that you need. But that doesn't mean that one approach doesn't exist. The way that I look at it is, why is AI and machine learning useful, very useful in imaging? Well, because there were billions of images that were able to be used to fine-tune models, machine learning, AI models for that purpose, and classify and get better at and perhaps replace human intervention or even be better than that at this point. Where else was AI useful? In text. Why is that? Because there is a huge amount of text well annotated that you can use to learn, and that's why it works in there. The other place in the drug discovery space where there is a little bit more effort is in trying to design molecules based on AI. And that has gotten reasonably far. But in terms of, getting to the point where AI can help on, providing the dose that you should be using, not yet. However, the part where it would be very useful and particularly of interest, one of the things that the AI does well is aggregate information. What would the reviewer at the FDA need to do? Aggregate the information that the sponsor company provides and identify, gaps or things where it requires more information. Why AI may be useful for that? Because once you identify an issue, you can check it. You don't rely strictly on AI to tell you what's going on. You can check that. And it's that checking, that human intervention that can be tremendously useful. So that application would be not as much about using AI in a completely disruptive, novel way, but actually using it to improve processes that exist today. Hopefully that answered your question.
Yeah, absolutely. Thank you so much. So it's basically trying to sort of not necessarily making models with AI, but making sort of using AI to solve tasks that would be difficult. Correct.
I mean, you could, in principle, build models. I know that there are folks that are trying to do exactly that. Is basically, reconstruct pathways, if you want, based on using AI. There are problems with that, but there were problems with you doing image analysis a few years back. They're not today. So.
Okay, well, if there are no more questions, Valeriu, thank you so much for the very nice presentation today. I really appreciate it.
Thanks, Tom. Thanks for the invitation.
Thank you. Have a great day, everybody, and happy holidays if we don't see you before then.